Yeah, so here we're going to see a full-scale bioreactor. This bioreactor is from a wastewater treatment plant operated by a utility in Canada. Here you see that anoxic, anaerobic, and oxide zone. Followed by fine bubble aeration. There you see where the influent pipe is located. You also have internal recirculation for NO3 recirculation, sludge recirculation, and your outlet pipe. So basically, we are doing this project together with this Canadian utility because their ultimate goal is to reduce their carbon footprint, so the greenhouse gas emission from operation to support their climate change mitigation strategy. And we are currently collaborating, and we are using the CFD N2O model to inform upcoming fieldwork to help them and support them in the next measurement campaign. So in this plant here in Canada is as a very big challenge with seasonal variation, because in winter temperature can go down. It can vary between -10 and -20 Celsius degrees. So it's a huge temperature difference that can occur along throughout the year. This is, for example, in this slide, we're looking at some N2O emission from two wastewater treatment plants in the Netherlands, operated by the wastewater utility Eindmaas, which kindly provided this graph. So basically, in the left graph, you can see that there is a clear pattern, a clear decrease of N2O emission during the summer, while in the second plan, this pattern is no longer that obvious. So basically, it's N2O emission seems to be correlated with temperature, with seasonal impact, and higher emission can occur in winter, but it cannot not be the same case for all the plants. So it's very case specific, and we are going to start a project with this utility to really investigate these issues further. So going back to the Canadian plant, here we see the root causes. So here first of all, we look at the heterotroph production rate, and we're looking at two different scenarios. One is at 11 degrees, and the other one is at 20 Celsius degrees. So two different temperature. First of all, the first thing that we can notice is that at 11 degrees, the production of heterotroph reduced. So basically, they slow down their activity. But actually, it's also important to mention that the production reduced, but also their consumption reduced. This is very important to highlight here. And then if we go to the heterotroph pathways, we can see, starting from the temperature at 11 degrees, we can see that the A1 process, the production kind of squeezed in between the anoxic and aerated zone. So the transition zone between anoxic and aerobic is gets reduced. But on the other side, we can see that there is some production by the A1 process in the final part of the reactor, where the N2O concentration are quite low. For the A2 process, we can see that the highest production is now reduced at 11 degrees, but their production is occurring throughout the whole aerated zone. So basically here, if we go and quantify the root causes, we would see that in this case, 70% of the N2O emission at 11 degrees are 72% is produced by the A1 process, and the remaining by the A2 process. Similar for 20 degrees. So what is very important here to see is that actually heterotroph in this case, they don't produce basically any N2O. And the reason is that they produce what they consume, what they are producing. And they are even consuming more than they are producing. So if we quantify the emission, we can see that in the end, at lower temperature, so at 11 degrees, the yearly emission and also the emission factor increases. Now we go to a mitigation strategy. So it's a common knowledge that reducing your aeration is an effective measure to reduce also the N2O emissions. So we are still in the Canadian plant, and here we compare a scenario with average aeration, and we see the N2O concentration in the liquid and in the gas. What we can see is that concentration increases in the anoxic zone, concentration in the liquid increases in the anoxic zone, and then what we can see is basically in comparing the two scenarios is that by reducing the aeration by 20%, actually our concentration in the liquid and in the gas increases. So this is the very first results. And why is that? It's because by reducing your aeration, you are also stripping less, which means that your N2O in the liquid gets accumulated, and therefore also your gas gets more concentrated. If we go a step further and we look at the root causes, again, we start with the heterotroph production, and we can see that with lower aeration, there is a bit more production of N2O, but actually there is also a higher consumption. So in the end, the net production by heterotroph is even negative. If we go to the other pathways, so the autotroph pathways, here we see that with lower aeration, the transition zone between anoxic to aerobic increases. So we can see that there is more production by the A1 process. And same happens also for the A2 process, because our ammonia lives a bit longer in the system, and then therefore you end up with enhancing this pathway. So again, quantifying the root causes, what we see is that in case of 20% less aeration, it's basically half and a half. So all the N2O is produced by the autotroph, and it's spread equally between the two pathways. With average aeration, we have slightly more production with the A1 process. Quantifying the emission, so it's from this graph, it is clear that reducing your aeration is an effective measure to reduce the emission, the CO2 equivalent emission, and also the emission factor is lower. However, it is also important to highlight here that it is an effective mitigation strategy. However, we could see some side negative effect. For example, the autotroph pathways increase their formation. So it's important to mention that there are some there is a need to fine-tune, to refine the mitigation strategy to counteract this side effect. For example, by controlling the aeration packages specifically.